Okay, welcome back to class. We are skipping a couple of chapters today, moving to chapter 6. And the reason why is because in chapter 6 is there's a little nugget in there. So I'm skipping to a section in chapter 6, not all of chapter 6, so that I can get nuggets out of it about enzymes. Since we talked about macroprotein and being one of the macromolecules, I'd like to go ahead and continue talking about enzymes, which instead of it being in chapter 3 with the rest of them, they tossed it over to a section in chapter 6. And so that's what we're going to do. Also, I'm skipping some of chapter 6 because they start talking about energetics, and a lot of that stuff about energetics is not going to be on your AP biology test. There'll be some grasp of concepts, but not a whole lot of testing over it, like things like exergonic and intergonic and Gibbs free energy. They won't ask you to calculate Gibbs free energy, they just want you to understand what the principles are behind it. So we'll be skipping that. We'll come back to it later, but we'll be skipping it for now. So you'll notice that your notes handout guide that you have is titled 6.4 on the top, and that's because that's the section we're skipping to get to enzymes and include that detail with the chapter 3 proteins, so that we're continuing from where we left off on proteins. Enzymes speed up metabolic reactions by lowering energy barriers. We'll talk a little bit more about that. But on your notes, it starts with enzyme. And so get this down, an enzyme is a catalytic protein. And then for catalyst is a chemical agent that speeds up a reaction without being consumed. But enzymes can also slow down reaction. So we call those inhibitors. And so we're talking about it speeds up metabolic reactions. These reactions would happen anyways, but enzymes help speed those up. And important to note here is that enzymes don't then become part of like a reaction equation. They are outside of that equation because the enzymes themselves are not changed. They only help speed that up by lowering and they also lower the energy barriers. That means that some reactions take more energy to get started. You have to introduce a little bit of energy just to get it started. For instance, like you strike a match and you light a candle, and then after that, it keeps giving off light and energy. But it took that striking the match and lighting it to begin with, so a little bit of activation energy to make it happen. So what an enzyme does is actually lowers the amount of energy it takes to get that reaction to start happening. And then hydrolysis, we talked about that already. Like if you have two sugar molecules that make up sucrose, like glucose and fructose, making up that sucrose molecule, hydrolysis of that by an enzyme called sucrase is an example of an enzyme catalyzed reaction. So that enzyme is involved in getting that reaction to happen faster, providing binding sites for the two glucose or glucose and fructose monosaccharides to come together and become this polysaccharide sucrose. Notice your sugars end in ose, and your protein, more specific, your enzymes end in ace, with the prefix that describes what it is that they are catalyzing. So sucrose, sucrase, see the tie in there? So you can see that beautifully illustrated here. You've got a sucrose molecule. It's all together. Add water together. So this is using water to lyse it, to cut it, right? And break it down. So then they were put to, so now it's separated out. It's glucose and fructose. And sucrase was the enzyme that helped provide binding spots before that. So let's. Now, if this was to work backwards, instead of hydrolysis happening here, if you're working backwards, then you're taking water molecule out of their dehydration reaction to make sucrose. Every chemical reaction between molecules involves bond breaking and bond forming. If you're bond breaking, that's going to release energy. If you're bond forming, that's going to require energy. The initial energy needed to start a chemical reaction is called the free energy of activation, or in other words, activation energy. So let's get that definition down in your notes. Activation energy is often supplied in the form of thermal energy, like I was talking about striking a match, that the reactant molecules absorb from their surroundings. This is why the sun is so important, because it's a source of thermal energy that helps with these reactions. If we had no sun, a lot of, well, all biological processes would begin to cease as we know of them, unless you, of course, you've adapted in some kind of way and you're one of these life forms that use chemicals as their source of activation energy, like some of these deep sea organisms. But in that case, again, if there was no sun, things would freeze. So that's a whole nother thing to talk about. So you can see in this diagram the whole chart. And this is showing that it took this much energy right here, took this much energy to get the reaction started. And then once that energy got started, it released all this energy. So you can see it took a little bit of energy to get it going, but it released a whole lot more energy. So you can think of this as, here I lit the candle. Now the candle is giving off light and heat. On your notes should say draw endergonic reaction and exergonic reactions on the graph. So we'll get back to this here in a little bit so you can get those two different types of reactions down. Enzyme catalysts, how do they speed up a reaction? Enzymes catalyze reactions by lowering the energy barrier. Enzymes do not affect the change in free energy, delta G. Instead, they hasten reactions that would occur naturally. So we're showing that here. Here's what it would normally be, the course of reaction with an enzyme. Here's what it would normally be. So here's the amount of energy it would normally take without an enzyme and the amount that it releases. Here is with an enzyme, it only took this much energy. So you could think of it as your helpful helperton, making the job easier. Many hands make light work, so to speak, and then enzymes and a little extra worker there, if you want to think of it as that analogy. So it lowers the activation energy, and you're able to get the same job done without having to expend a lot of energy to make it, as much energy to make it happen. The reactant that an enzyme acts on is called the enzyme's substrate. So let's get that down for the definition. The enzyme binds to its substrate, forming an enzyme substrate complex. That down for a definition. So what we're describing here is something that the enzyme binds to its substrate, and then when they're together, it's enzyme substrate complex. I like to think of it kind of like that. So going back to drawing that endergonic, exergonic reactions, if you'll put that on your chart there. Endergonic, so ender, think of the prefix endo versus going into, and exiting. Endergonic, you'll see a lot of energy had to go into it, and it only releases a little bit of energy. So think of things that are getting really colder because they're absorbing heat from you. So like these little icy hot packs where you get the chemical reaction going by breaking the bonds and stuff like that, and then that pack gets really cold and what it's doing is drawing heat out of your body. So that would be an endergonic reaction because it has to absorb a lot of energy and only gives off a little bit. And then exergonic, you see exiting here, a little bit of energy is required, so striking a match, but then a whole lot of energy is released in that. So this is exergonic. And then you have, it says write and the equation with an enzyme substrate reaction. So that's actually back in section 1 where you've got a molecule, enzyme is involved, and you get reaction 2. Enzymes involved, reaction 3. Enzymes involved, product. If one of these, the point with this that I want to make is if one of these enzymes doesn't work, like let's say enzyme number 2 doesn't work, then that doesn't affect enzyme 1 and making its reactant, but it does stop 2 and 3 from making, it does stop this particular molecule from being formed, this square here representing some kind of molecule. So molecule 1 and molecule 2 will get made, but then molecule 3 won't if enzyme 2 is denatured or doesn't work the way it's supposed to. So going back to our definitions, the active site says describe the following two. So the active site is the region on the enzyme where the substrate binds. We'll get that down. The enzyme's specific. So this part I would make a definite side note either at the top or the bottom of your page, because this is definitely, this concept anyways is definitely tested on many different ways. Enzyme specificity results from the complementary fit between the shape of its active site and the substrate shape. Sorry, I can't speak right now. The enzymes are, what this is saying is the enzymes are very specific to what they'll bind to. They're like lock and key. The key doesn't work well without the right lock, and the lock doesn't work well without the right key. They go hand in hand. So that's why we name enzymes the way that we do. We name the prefix after what the substrate is, and the suffix is ACE because it's the protein for that specific substrate. So they're very specific and don't work with other things. Enzymes change shape due to chemical interactions with substrate. There's other ways that we can make them change shape, and when they do change shape and get bent out of place or denatured, then they can't work the way that they need to because what was supposed to fit in there doesn't fit in there like it's supposed to anymore and doesn't work like it's supposed to anymore. The enzyme. But when they work complementary like this, the enzyme changing shape due to the chemical interaction with the substrate, then we have something called the induced fit of the enzyme to the substrate. Get that down for induced fit. So the induced fit of the enzyme to the substrate brings chemical groups of the active site into position that enhances their ability to catalyze the reaction. Otherwise, you have these chemicals that eventually will run into each other, but there's no, how shall I put it, matchmaker. You can think of an enzyme as a matchmaker saying, hey, look, I know you and I know you. Hey, why don't y'all get together? I think y'all will fit perfectly together and work and interact. Well, that's what an enzyme does. It helps make that happen faster than it would before. So you can see a picture of that here. You've got this active, here's your big enzyme made of all these folded proteins. And then you've got this perfect spot for this substrate, this active site for the substrate to fit in. And once that substrate is in there, it kind of creates this enzyme substrate complex where you kind of makes its own different kind of complex. Again, the enzyme, everything about the enzyme is there. They're not chemically reacting together. So it's not part of the reaction itself. It's just allowing a reaction that would have happened to happen faster. Okay, so catalyst and the enzyme's active site. In an enzymatic reaction, the substrate binds to the active site of the enzyme. The active site can lower an energy barrier by these four ways: orienting the substrate correctly so that they line up just right and can bind together, straining the substrate's bonds so that they can break those bonds more easily, providing a favorable microenvironment, and covalently bonding to the substrate. So you can see here, you've got, let's pretend like this is glucose and fructose. And right now, the way that they are oriented, they might eventually bump into each other and be like this. Or maybe this flips around and bumps into there and they don't work together. They weren't in just the right situation to bind. But when they both run into this enzyme, they fit in these spots just like a key and a slot there, or a key and a lock there, and provide this perfect way of fitting them two together so that that reaction happens and happens more quickly. So then this becomes what we call the enzyme substrate complex. The substrates are converted into products. So now it's a new chemical reaction has happened now, and then they're released from that enzyme now that the chemical reactions happen, and products then can be released. The active site's still available. Like I was mentioning, the enzyme itself's not changed in the chemical reaction. These are, but not the enzyme itself. And so it continues to do its job. Active site's available for it to happen all again. So it's almost like a matchmaking machine. It is a matchmaking machine. Things that can affect the local conditions of that enzyme, and enzyme's activity can be affected by the general environmental factors. We kind of mentioned these before for the test. We can change the temperature, make it hotter or colder, denaturing it. So imagine, that's why, for instance, we would keep cut apples on ice so that they don't oxidize as fast. Or changing the pH, maybe spritz a little bit of lemon on there, changing it to more acidic, and so it denatures those enzymes, keeping it from oxidizing, apple from oxidizing. Chemicals that and then there's also chemicals that specifically influence the enzyme. So let's break down temperature and pH. Each enzyme has an optimal temperature in which it can function. Each enzyme has an optimal pH in which it can function. Optimal conditions favor the most active shape for the enzyme molecule. You take them outside of their optimal range and you either denature it or change its shape, and it doesn't work as well. So you can see that here, starting to heat it up or starting to change its pH. Let's get this down in our notes. Cofactors are non-protein enzyme helpers. Cofactors may be inorganic, such as metal in ionic form, or organic. For a coenzyme, it's an organic cofactor. Coenzymes include vitamins. So this is why vitamins are so important to help you with your digestion. They're not enzymes, but because again, it says non-protein enzyme helpers. So they help the enzyme. They're not an enzyme. On the other side of that page, you have competitive inhibitors. They bind to the active site of an enzyme competing with the substrate. So get that down. So you've got that here. Something is in the way of that active site, and so this substrate can't get in there. So that would be competitive inhibitions. This is what it would. This would be an ideal situation. Normal binding. This would normally fit in just right, but with that in the blocking the way, it can't. And then non-competitive inhibitors bind to another part of an enzyme, causing the enzyme to change shape and making the active site less effective. So you can see right here, it's not binding to the active site, like right here, the active site's right there, the active site's right here, but look what happened to the shape of that active site. It changed it. It's like we stuck a wedge in right there, making this end of it pinch closer together or change shape, and therefore this doesn't fit like a lock and key anymore. It doesn't work like it's supposed to. It's not on the activation site, so we call it non-competitive, but it's still inhibiting it, so it's a non-competitive inhibitor. So examples of inhibitors include toxins, poisons, pesticides, and antibiotics. So we kind of have an understanding of what those kind of things do, and now kind of know why they are effective. So some toxins and poisons or pesticides may not necessarily kill a bug directly, but would keep the bug or plant or whatever from being able to have access to simple metabolic processes that would cause it to survive, or getting the hormones being able to do what they need to do, and all kinds of different processes that need these enzymes to work the way they do. So when they don't, it equals death for them. So the question on your paper says, how do active energy barriers and enzymes help maintain structural and metabolic order of life? So write this down. I'll say it out slowly. Activation energy barriers prevent the complex molecules of a cell, which are rich in free energy, from spontaneously breaking down to less ordered, more stable molecules. Okay, you need to pause and go back to that. That's fine. A couple of other things, tying, we always want to kind of tie evolution into this as one of the big ideas. Enzymes are proteins encoded by genes. The gene, we've discovered that it's the genes that code for life, that code for making the proteins, and not the proteins, so not the other way around. Changes or mutations in genes lead to changes in amino acid composition of an enzyme. So that can change that enzyme, sometimes even making it where it doesn't work like it's supposed to. Altered amino acids and enzymes may alter their substrate specificity. It's a hard word to say. Under new environmental conditions, a novel form of an enzyme might be favored. Okay, we'll end in 6.5, last section here. Chemical chaos would result in a cell's metabolic pathways if they were not tightly regulated, going back to answering our question earlier. A cell does this by switching on and off the genes that encode specific enzymes or by regulating the activity of enzymes. We'll get more on that later on, not today. Get this down, allosteric regulation may either inhibit or stimulate an enzyme's activity. Allosteric regions occur when a regulatory molecule binds to a protein at one site and affects the protein function at another site. Most allosterically regulated enzymes are made from polypeptide subunits. Each enzyme has active and inactive forms. The binding of an activator stabilizes the active form of the enzyme. The binding of an inhibitor stabilizes the inactive form of the enzyme. So you can see it here. Much like a non-competitive inhibitor, it wedges itself in there and changes their shape, making it in an inactive form. Substrate comes in, fitting in perfectly, making the rest of them say, I'll be that shape too, or stabilizing form. So that's cooperating, whereas this is more of an inhibiting. Sorry, I should refer to this. Here's the inhibitor fitting in there, changing them the wrong way, whereas this one actually makes it work better. It's wedging in there, but keeping them from moving around too much. So it's actually stabilizing it better on this one. So when I was mentioning inhibitor, this is the better example. This is the example I should have been using. This down, cooperativity is a form of regulation that can amplify enzyme activity. One substrate molecule primes an enzyme to act on additional substrate molecules more readily. Cooperativity is allosteric because binding by a substrate to one active site affects the catalyst in a different active site. And then feedback inhibition. Get this down, the end product of a metabolic pathway shuts down the pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. So later on, we'll talk about how do cells, how does the genetic code, how is all of that regulated? So you can see here some kind of regulation. You've got an active site. It falls into that active site, creating this enzyme substrate here. Then a series of enzymes and intermediate enzyme substrates and products happens, creating this in the end, and this actually becomes the inhibitor. Would be a non-competitive inhibitor type situation. Isolene binds to the allosteric site, and you see how that kind of changes here. So that's feedback there where it's stopping this continued process of making more of these. So if there's enough of these around, it can stop the process. So answering your question, what role do allosteric regulation and feedback inhibition play in the metabolism of a cell? It helps to regulate that. It helps to regulate the whole process. Not make too much of a product, or not make too little of a product. And then specific localization of enzymes within the cell. Structures within the cell help bring order to metabolic pathways. Some enzymes act as structural components of membranes, so they're embedded inside those. Can be embedded inside those membranes in cells. In eukaryotic cells, some enzymes reside in specific organelles. For example, enzymes for cellular respiration are located in the mitochondria. Example here, and they're in specific spots, and so that a whole metabolic pathway can form. And we'll talk more about those when we talk about cells later. So we'll end with this. How are the rates of enzyme reactions affected by environmental factors? Well, we talked about them already. We talked about pH. We talked about temperature can change that. But then there's also not having enough substrate or not having enough enzymes. So a limit on either one of those also would affect the enzyme rate. Thank you and have a good day. Be sure to turn in those notes.